It's just indeed my pleasure to talk to you today about some of the progress in hepatitis B cure research. Now, some of this you've already heard, so I have retailored my talk a little bit after Professor Ewan's talk yesterday and some of the other talks so that we're not overlapping too much. So I think that's important. So that's my disclosure. So the structure of the presentation, and again, we've heard a lot about the global burden of hepatitis B, so I won't dwell too much on this, or the natural history and the current treatments. But I will talk about the definitions of cure and some of the new therapeutic agents, perhaps some of the ones that Professor Ewan didn't elaborate on so much. And then I'll talk to you about our ICE-HPV, or the International Coalition to Eliminate Hepatitis B Global Scientific Strategy, which we released and was published in April. So we all know that more than 2 billion people across the globe show evidence of hepatitis B infection, and we have a chronic load of 257 million people, many of whom reside in our Asia-Pacific region, 120 million people in Asia living with chronic hepatitis B, and also 70 million people in Africa. It's a huge, huge burden. 887,000 deaths every year. And of course, as we've learned a lot these past 2 days, hepatitis B causes liver cancer. Liver cancer is the third most common cause of cancer mortality. It's the fifth most common cancer globally, but the third most common cause of mortality, largely because we diagnose it so late. We don't have effective biomarkers really for liver cancer, something we urgently need. In addition to cure research, we need research on biomarkers of liver cancer, and that is actually an area that's very dear to my heart. And of course, our current vaccine, we've learnt a little bit over the last 2 days, we have an excellent prophylactic vaccine, but it has no effect at all on existing infections. And it's a little bit of a problem because a lot of people, particularly funding organisations, think that because hepatitis B has a vaccine, the problem is solved. But we in this room all know that this is just not the case. And of course, the current treatments reduce but do not eliminate the risk of liver cancer. So our current treatment paradigm, our first-line drugs of entecavir and tenofovir, the direct-acting antivirals, long-term, not finite, lifelong treatment. But HBs antigen loss is very, very rare. And of course, the old immune modulator of pegylated interferon says here a response rate of 25%, but really it's very genotype specific. It really only works reasonably well in genotype A patients and perhaps genotype B in Asian patients, largely ineffective in genotype C, and very, very poor response in genotype D. And since many countries don't genotype their patients, the response to interferon is going to be largely unknown. So the current antiviral treatments are very, very effective in lowering the viral load, but they're not cures. Okay? In fact, the replicating, when they lower the viral load, I'll show you in a moment, when they lower the viral load but below the lower limit of detection, that replicating virus that's there is actually still infectious. And these treatments do not impact the integrated DNA. We learnt very eloquently yesterday about the importance of integrated DNA expressing the S antigen in the setting of E antigen negative disease. And of course, as I've just said, the therapy is lifelong and doesn't eliminate the risk of liver cancer. And this study from Patrick Marcel and published in Hepatology in 2014 shows that a large number of patients on tenofovir still had virus present at the lower limit of, below the lower limit of quantification, and in fact the lower limit of detection. And some studies recently published or presented at EASL show that when the serum from the patients where the virus was present at the below the limit of quantification, when that serum was put into mice by hydrodynamic injection, it proved to be infectious, and the virus replication was restored after some time. So this very, very low level of virus, even though it's below the level of quantification, is actually infectious. This has some ramifications for chronic hepatitis B. So can we cure chronic hepatitis B? And of course the cures for hepatitis C are amazing, as little as 4 to 8 weeks of treatment. And this has in fact raised expectations for hepatitis B cure. But they're very, very different viruses, as we all know. Hepatitis C replication is only in the cytoplasm. Once that virus is gone, there's no reservoir, the virus is gone. But hepatitis B is a little bit more tricky. It has the nuclear, the cccDNA, the covalently closed circular DNA phase, as well as the cytoplasmic phases to its life cycle. And it's this nuclear reservoir, this mini chromosome in the nucleus, that makes it so difficult to eliminate. It's a constant source of the viral proteins, the viral messenger RNAs, and subsequently the proteins, but also the reverse transcription, the template needed to subsequently make the viral DNA. So unless we can get rid of this cccDNA, we're never going to completely eliminate hepatitis B. So we talked a little bit about the definitions of cure. There's really two definitions in circulation at present, and these are under constant discussion because there's a lot of feedback from the hepatitis B affected community that the term functional cure doesn't mean a hell of a lot to patients. You go to a doctor and they say, you're functionally cured, and I say, what does that mean? Like, what's a non-functional cure? Okay, so the patients really want to work with us to get better definitions. But functional cure, as we learnt yesterday, is really sustained durable S antigen loss, with or without anti-HBs. There's a lot of evidence you don't need anti-HBs to maintain HBs antigen loss. And of course, the cccDNA is still there, but it's not going to be transcriptionally active, so it's silenced. But complete cure, which is really the holy grail of hepatitis B curative research, it's the same as functional cure, but we've got rid of that cccDNA or mini-chromosome reservoir. And so the major barriers to hepatitis B elimination, now, I'm a virologist, but it's not all about the virology. As we've learnt this morning, the immunology is going to be incredibly important to curing hepatitis B. It really is going to be a two-edged sword, a two-edged approach, if you like. We've got to target the virus, and we've got to enhance the immune responses in those people who have chronically exhausted immune responses, be they T or B cells. So, and again, it's this generation of this cccDNA reservoir, which we have here. So that following the infection of the virus enters the cell via the N TCP receptor, and then it is transported to the nucleus where the relaxed circular DNA is converted to this covalently closed circular DNA. And really, I'm going to focus on the new treatments that are targeting the cccDNA and also the entry inhibitors, because Professor Ewan talked about a lot of the other approaches in his talk yesterday. So HPV is currently incurable, but over 1 billion people have resolved an acute infection, okay? And we do see functional cure in a small number of people each year, small number of treated or untreated individuals, 1 or 2%, either spontaneously or on treatment. So in theory, if nature can do it, why can't we do it? Okay, so a functional cure is really a realistic goal, and the pharmaceutical industry agrees with this, and billions of dollars is now being poured into hepatitis B curative research, which is really, really wonderful. And the key discovery in hepatitis B in the last 10 years has really been the discovery of the N TCP entry receptor in 2012 by Professor Wenwei Li and his group in Beijing. So we now can set up systems where we can put the receptor into cells or even into small animals like mice, and we can infect those cells, and we can just look at the whole virus replication cycle, because it's only through infection that you generate or largely generate the cccDNA. So with this discovery of the receptor really did change our ability to study hepatitis B. So there's numerous approaches for treatment, and some of which may well be curative, targeting different stages of the replication cycle. And again, Professor Ewan talked a lot about a lot of these yesterday, and there's many of these are in various stages of clinical trial. I'm going to talk a little bit about the entry inhibitor, and I'll talk about a new molecule from Roche targeting the cccDNA, which is very exciting. So the cure pipeline, as I mentioned, is going to require direct-acting antivirals and immune therapies. I'm a big believer it's going to need a dual-pronged approach. And we'll talk now about the viral entry inhibitors and the cccDNA targeting. So the most recent and most widely known entry inhibitor is Melphalan. So the way hepatitis B, for those of you who don't know, is that the pre-S1 region of the large surface protein has a peptide which binds to the N TCP. Melphalan is an analogue, if you like, of that peptide and prevents binding of the hepatitis B to the N TCP receptor. And some results presented very recently at the last EASL meeting, and also again at the hepatitis B meeting in Melbourne only 2 weeks ago, showed a remarkable response that S antigen loss was achieved in 27% of patients treated with Melphalan. So this is very, very exciting. Now, the mechanism for this is still a little bit unclear in my mind, but it's likely to be preventing the reinfection of cells or preventing the infection of cells and the generation of cccDNA, which produces the S antigen. Excuse me, folks, I've got a little cold. Okay, but this is very exciting. I'd love a water. Yeah, thank you. Or some sake even, maybe just water. Thank you. Thank you. So this is very, very exciting though. Goodness me. I did not drink much last night, I assure you. I said no to the waiter a number of times. Anyway,
and I can witness that. I was sitting opposite him.
Yeah, I can't get rid of it. So anyway, 27% S antigen loss is incredible. Okay, so this is very, very exciting. But we've got to target the cccDNA reservoir. Now, a number of people have tried, we've all heard of CRISPR and these direct-acting approaches for targeting DNA molecules, and there have been attempts at using CRISPR to target cccDNA, both in cell culture and also in mice, and you can directly target cccDNA, but there's going to be problems with delivery of getting these sort of molecules into the human liver, and off-target effects, because hepatitis B integrates, or parts of the genome integrate. If you target those, you're targeting your own DNA. Okay, and the other problem with CRISPR, at least earlier iterations of the technology, it doesn't eliminate the cccDNA. It chops a little bit out and then it reforms into a new molecule. We're making a new virus, folks, if we're doing that. Okay, so there's a lot to work through with CRISPR and those sort of approaches, but the latest approaches to CRISPR do completely eliminate DNA molecules. They chop it up completely. Again, if the virus is integrated into my DNA, I don't want it chopped up completely, so we have to do a lot of work looking at that. But Roche presented at EASL recently in Vienna a molecule which, and I don't know the mechanism at all, I don't think that's been disclosed yet, but a mechanism, a small molecule that directly targets cccDNA. And they infected primary human hepatocytes and they used what they call their mini-circle molecule in mice. And that's where you hydrodynamically inject, like a cccDNA, like a plasmid, that represents cccDNA into the mice, and it generates a molecule which you can directly target. And just showing you the effectiveness of this molecule here, of this small inhibitor, and this here shows that as increasing dose, this is in the primary human hepatocytes, just looking at a southern blot, the cccDNA is disappearing. And then in the mice, the S antigen on treatment was the cccDNA, so S antigen and also the viral load were decreasing. Very, very promising. I have no idea of the mechanism, but this is the first time we've seen a drug in an early stage development phase that can directly target the cccDNA. Obviously, we need to know how it works and if there are any off-target effects and all that sort of thing, but it's very, very exciting. And of course, there's a lot of work now, a lot of interest in the SMAC5/6 pathway, which is critical for the regulation of transcription and the maintenance of the cccDNA pool. And the interaction of HBx with the DDB1, the damaged DNA binding protein, is proving to be a very interesting area where people, if you can target that interaction between HBx and DDB1, it interferes with the SMAC5/6 and you may be able to repress cccDNA transcription. So there's been a number of presentations recently on this very thing. There was an invited talk at AASLD in 2018 looking at a novel chemical for disrupting this interaction, and there were posters at ILC in Vienna, and then we had 2 posters recently at the International HPV meeting in Melbourne last week. So there's a lot of interest in the field in targeting this DDB1 HBx interaction. You're not targeting the DDB1 itself, that's a host protein, but you're targeting that interaction. There's a very specific binding site between HBx and DDB1. And it turns out too that DDB1 binds to DDB2, and DDB2 might be what's binding to the cccDNA. And Heito Gao's group in Pennsylvania or in the USA is working on that. So there's lots of work in that field. So in terms of immunotherapies, I'm not going to talk too much about immune therapies, but we all know that we need to restore the immune response, and we learnt this morning. We need to restore the immune response to those of a person who either acutely resolves an acute infection or clears a chronic hepatitis B infection. But in a chronic hepatitis B infection, people have a weak immune response and exhausted T cells and a poor B cell response. So there's a lot of work in the field, and we learnt about some of these this morning, looking at PD-1, looking at therapeutic vaccines, engineering T cells, but also boosting the innate immune response. We learnt yesterday the importance of triggering innate immune responses to clearing viral or treating viral infections or targeting viral infections. So again, hepatitis B is going to need a multi-pronged approach, and one particular approach being undertaken by Antonio Bertoletti's group and group in Singapore is looking at engineering non-lytic lymphocytes, so not clearing through cytolytic clearance, but actually activating the ApoBec3 pathway, which has been shown in a science paper by the Protzer group to reduce cccDNA and target it for deamination. So by stimulating T cells and stimulating this pathway, and these are resting T cells, and I believe me, as a non-immunologist, I don't quite understand the technology, but the field's quite excited about this study as well. So it's going to require multiple approaches, targeting direct-acting antiviral approaches and immune modulatory approaches to target different aspects of both the host immune response and also the viral replication cycle. So and many of these approaches are in phase 1 and phase 2 clinical trials, which is really, really exciting. I think there's now 30 different drugs in phase 1 and phase 2, early phase 2, which is amazing. So as I said, there's a huge amount of interest in the field. for hepatitis B. And so, but we haven't talked much about combination therapy, and that's perhaps the next step for the pharmaceutical industry, is which drugs are they going to combine to cure hepatitis B? And, you know, are the industry going to work together if somebody's producing a capsid inhibitor and if somebody else is producing a cccDNA inhibitor and somebody else is producing an siRNA? Are they all going to work together, combine their drugs to cure hepatitis B? Or is the one company going to have all of the arboretum to do it themselves? And there's a lot of discussion, and the industry are working very closely together. It is quite interesting and exciting to see how collaborative the industries are. But there really is an urgent need for a coordinated approach to hepatitis B cure, and prior to 2016, our efforts weren't coordinated at all. The HIV field has been coordinating cure efforts since 2010 through the International AIDS Society, but we don't have such a society for hepatitis B. And given that Hep B is a global problem, we really thought that we need global collaborations to cure hepatitis B. So in 2016, we formed an organisation called ICE-HPV, the International Coalition to Eliminate Hep B. And we aim to fast-track the discovery of a safe, effective, affordable and scalable cure to benefit everybody living with chronic hepatitis B, including kids, because this is a disease of children, and people living with comorbidities. And we intend to contribute to the elimination of chronic Hep B as a global public health challenge. We work with a lot of stakeholders. We work with a lot of community, a lot of people living with chronic hepatitis B, a lot of world leaders in the field. We work with very, very closely World Health Organisation, World Hepatitis Alliance, and others. And we set up working groups in virology, immunology and innovative tools, and this was mirrored on the approach put together by the HIV field. And those working groups put together a brainstorm for over 2 years to come up with a global scientific strategy on what's required for hepatitis B. We published this in April and we had a press conference launching it at EASL in Vienna. Basically the two strategies that were identified were we need to cure infection, focus research on curing infection without killing HPV-infected cells, so getting rid of that cccDNA reservoir, but also inducing the immune control to safely eliminate the infected cells. And I won't go through the whole paper, this has now been published, but the strategies are detailed on there. If anyone would like a reprint, please send me an email. But the immediate and future actions required to achieve HPV cure, the ICE-HPV strategy is, firstly, we need to increase funding. Okay, we need a lot more funding for hepatitis B research, not at the expense of funding for HIV or hepatitis C, but we need to increase the pool of funding so there's more funding for hepatitis B research. And we're very, very excited that only last week or the week before in Lyon, the Global Fund replenishment meeting for the first time included hepatitis virus elimination in their agenda. And ICE-HPV played a key role in getting that on the agenda. So we look forward to the outcomes of that. To be part of the Global Fund as elimination of viral hepatitis would be amazing. We need to concentrate on the discovery of interventional strategies that will permanently silence or eliminate that cccDNA and stimulate specific antibodies and T cells that will prevent viral spread to uninfected cells and mimicking the spontaneous resolution of hepatitis B infection, be it acute or chronic. And we need to establish repositories of standardised reagents and protocols that everybody around the world can access. We don't have this for hepatitis B yet. Okay, the HIV field can get on the phone, ring up the NIH and say, I would like a peptide library, or I would like a clone, I would like an antibody, and it'll be delivered free to their laboratory. We don't have that for hepatitis B, but we're getting it. And so ICE-HPV initiated this, and one of our governing board members, Jake Liang, works for the NIAID at the NIH, and they are establishing right now a reagents repository for hepatitis B that researchers all over the world are going to contribute to, and it's going to be available to everybody around the world for free. Okay, I myself am putting in clones and some antibodies. So the field is currently working out exactly what needs to be in this repository. We had a survey at our recent Hep B meeting in Melbourne, but this is going to be a hugely important resource. And we're also setting up a protocols database in parallel, okay, because you need to know how to use the reagents, okay? So that's being set. It's on the ICE-HPV website if anyone wants more information about that. So how far are we from cure? We've made considerable advances in the past 5 years. There's huge interest in the pharmaceutical industry. We now have global coordinated approaches, and research funding is increasing. There was a mono infection call for hepatitis B curative research by the NIH in December last year for the first time, $4 million. That's fantastic. So these factors are going to contribute to a marked increase in Hep B functional cure rates in the next 10 years. I'm not going to put a date on when we're going to have cure for hepatitis B, but we are going to get there. In Melbourne at the recent meeting, hepatitis B meeting, we signed what we called the Melbourne Declaration to try and improve funding and work and interest in hepatitis B in Victoria. And basically we called for a substantial increase in government and industry funding dedicated for increased testing and molecular diagnostics, treatment and curative hepatitis B research to facilitate equitable, affordable and universal access to hepatitis B cure within the next 10 years. This was signed by a number of institutes, the Doherty Institute where I work, the Burnett Institute where Margaret works, the International Hepatitis B Foundation, the International Hepatitis B Meeting and the World Hepatitis WHO Collaborating Centre for Viral Hepatitis, among others. So we think this is going to be a terrific declaration for us to then use as a starting point to try and generate more interest and funding for hepatitis B. So 2015 was the 50th anniversary of the discovery of the Australia antigen, which we now know as the envelope protein of hepatitis B. And we at ICE HPV and all our partners and friends are determined it's not going to be another 50 years before we have a cure. So I thank and acknowledge all of these organisations and people. I thank you for your attention and invite some questions.